So thank you. Okay, it's next up is Professor Velandai Srikanth. So Srikanth, again, is known to many of you. I've known him for a very long time. Sorry, Srikanth. He is currently Professor of Medicine at Peninsula Health and Monash University, and he's leading cohort studies in the field of metabolic health and dementia with a big interest in diabetes, type 2 diabetes. Srikanth's had a long interest in cognition and stroke. He did his PhD in it. He's led many excellent studies looking at cognitive outcomes as well as other risk in the elderly, including falls. And today he's going to talk to us about some of his work in type 2 diabetes. So this is type 2 diabetes in the brain, underlying pathways. Thanks, Srikanth.
Thank you, Amy. Can everyone hear me? Good, great. So this is really nice to be invited to speak here at this symposium. I've got 15 minutes. I've got too many slides. I'm going to try and whip through them because I'd like to tell you a story today. So we've been working, my collaborators and I have been working in the field of type 2 diabetes and dementia for the last 10 years, and we've got some insights into what pathways might be important. These are my disclosures. The outline of my talk, I'm just going to briefly tell you about dementia, which you all know about, but more importantly talk to you about type 2 diabetes really briefly, and then go straight into the link between type 2 diabetes and dementia. What I'm really going to concentrate on today are the insights from neuroimaging studies that we've performed looking at the question of what the pathways might be, whether neurodegeneration is important, whether vascular disease is important, or is it a combination of the two. Dementia is important. That's all I'm going to say about dementia. Type 2 diabetes is also quite prevalent in the population. About 80% of people with diabetes have type 2 diabetes. The worldwide prevalence is really high. It's characterized by insulin resistance, which might become an important mechanism in the whole scheme of understanding dementia pathogenesis. It is also associated with a number of biochemical and other mechanisms such as adiposity, lipid dysregulation, glycation, chronic inflammation, all of which might be important in linking diabetes to the brain. And then we know that diabetes is commonly known for its cardiovascular complications, microvascular disease, macrovascular disease, blood pressure-related problems, and as Amy pointed out, potentially cardiac problems. The risk of future dementia and type 2 diabetes is well established. There have been a number of cohort studies in the past which have established about a twofold increase in the risk of dementia after type 2 diabetes. If you look at quite a unique population-based twin study, which is a discordant pair study, looking at people with dementia, the co-twins without dementia, looking back at the exposure of diabetes, there is that risk which is confirmed. The important thing here is that when you stratify by the time of diabetes onset, the risk seems to be higher when diabetes onset is at midlife rather than later life. So the pathways, vascular or neurodegenerative. There are three specific studies that I'd like to draw my conclusions from. The first is the Melbourne T2D twin study. The second is a larger cohort study in Tasmania called the Cognition and Diabetes in Older Tasmanian study, and then data that we borrowed from the Alzheimer's Disease Neuroimaging Initiative, which is a very large clinical study in the United States. The photographs of the people that you see on the right are the PhD students who have been associated with these studies. So the MED study. This is a discordant pairs study. So we have people with type 2 diabetes and their co-twins without type 2 diabetes, and we're comparing them for several brain outcomes. And the questions that we asked is, in middle-aged twins, is type 2 diabetes associated with a reduction in specific cognitive abilities, brain structural changes, and is this independent of vascular disease? What about functional brain activation after a memory task? And are there differences in perfusion and metabolism? So it's a co-twin study, discordant for type 2 diabetes. Initially we had about 38 pairs, age just over 50 years, about mean age about 60, derived from the Australian Twin Registry. We threw a number of investigations at them, which included a comprehensive cognitive battery, a fairly significant set of MR imaging, and now more recently PET studies, and we did analyses which looked at within pair differences. This table is describing the sample characteristics, and you'll find that the most important thing here is that the twin with type 2 diabetes has a different phenotype to the twin without diabetes, confirming the difference in disease presence. The interesting thing we found in this group is that there were absolutely no differences in cognitive scores. So how they performed the cognitive scores, and this is about a mean age of 60, were exactly the same. We didn't find any differences in the standard measures of brain volume or vascular lesion volume, but bear in mind that the actual burden of vascular disease, as we can measure it now, was small. But the interesting thing here was how they approached the functional brain memory task. These panels show you the differences between twin pairs in all pairs, 22 pairs. These are monozygotes, these are dizygotes. The blue areas are those areas of decreased activation in type 2 diabetes compared to the twins, and the red areas are those of increased activation. And the interesting finding was that there were some clear-cut areas of reduced activation in areas such as the cingulate cortex. But the more interesting thing was that there were much more areas that the person with type 2 diabetes had to recruit in order to reach the same cognitive endpoint as their co-twin. 3 years later, we are still completing the analyses of further tests. We do find that there is reduced attentional abilities in the diabetic twin. Still no differences in brain volumes. There is reduced white matter integrity in certain areas. No differences in ASL measured cerebral perfusion. What we're finding is a striking reduction in FDG uptake, suggesting that glucose metabolism in the brain is altered. What about older age? These are twins who were in their sort of late middle age. What about older age? And this is the CDOT study that I spoke to you about earlier. These people were aged about 70 years. We have about 700 people with and without type 2 diabetes with several measurements. The questions we asked in these people is that, is type 2 diabetes associated with poor cognitive function or decline? Is it associated with greater brain atrophy independent of cerebrovascular disease? And what is the role of these brain changes to the observed cognitive changes? These papers have been published. The primary paper, which looked at the cross-sectional differences in brain imaging and how it related to cognitive performance, was published by Chris Moran in 2013. And more recently, we have a publication in Diabetology looking at the longitudinal changes. Again, the sample characteristics shows that differences are consistent with the diabetic phenotype. And we found at baseline that there were some differences in cognitive function in these people, predominantly in visual spatial planning, a bit in speed. As you adjusted for more variables, this difference in visual spatial planning persisted. When we looked at brain imaging markers, we found that there were significant associations with hippocampal volume, total grey matter volume, and white matter volumes. Yes, there was now an association with the presence of infarcts and MRI, but not with the other markers of vascular lesions as we know them to be. The regional distribution of brain atrophy or brain volume loss was fairly similar to what you might see in someone with Alzheimer's disease. Now, I'm not trying the board to say that this is Alzheimer's disease, but there are similarities. When we looked at mediation of the cognition type 2 diabetes relationship, so this is the beta coefficient and 95% confidence interval. Here we have the adjustments. This is the initial model adjusted for age, sex, and mood. When we put in grey and hippocampal volumes, that took away that association by about 40 to 50%. When we put in infarcts and the other measures, there was hardly any change in the betas, suggesting that this association is being driven by loss of grey and hippocampal volume. When we followed these people up for about 5 years, we do see that there are reductions, or there's a greater rate of change in cognitive abilities, primarily in verbal fluency and verbal memory. There was a trend towards increase in ventricular volume, didn't reach statistical significance. But although total brain volume was lower in type 2 diabetes at baseline, there was no difference in the rate of change over this time period that we observed. What about in much older people with dementia? This is from the ADNI study. For people who don't know what the ADNI study is, it's a multi-center clinical study recruiting people with Alzheimer's disease, MCI, and normal cognition to look at the risk factors for progression from MCI to Alzheimer's disease. We derived data from these samples to look at the question, in older people with AD, MCI, and normal cognition, is type 2 diabetes associated with markers of neurodegeneration and measures of A, beta, or tau? And is the rate of cognitive decline different? And what are the mediators? First thing we confirmed is, yes, there is a relationship between type 2 diabetes and reduced cortical thickness, but this was not modified by the presence of either Alzheimer's disease, MCI status, or normal cognition. There's clearly no relationship between type 2 diabetes and PET amyloid binding. We found that type 2 diabetes was associated with CSF tau and p-tau, but not with CSF A-beta. And when we followed these cohorts up longitudinally over 6 years and applied structural equation modeling to look at causal pathways, we found an indirect pathway between type 2 diabetes via its effect on baseline cortical thickness towards the rate of change in cognitive function. We also found that in the presence of greater education at baseline, that effect of type 2 diabetes on cortical thickness was modified or ameliorated, raising the question of, is it cognitive and brain reserve that are making a difference here? So if you put all that together, in late middle age with well-controlled type 2 diabetes in the absence of significant cerebrovascular disease, we found associations between type 2 diabetes and brain activation patterns. 3 years later, we see early changes in cognitive function. We see some changes in white matter integrity, widespread reduction in FDG uptake, but no changes at this point in MRI measures of volume or perfusion. If you look at older people with reasonably well-controlled type 2 diabetes, it is very strongly associated with smaller brain volumes and infarcts. The T2D brain volume association explained a substantial portion of the link between type 2 diabetes and cognitive function, rather than the T2D infarct association. Type 2 diabetes was associated with greater levels of CSF p-tau, but is not associated with brain or CSF beta amyloid, and that's independent of AD, MCI, and normal cognition diagnosis. And there's some interesting data to suggest that brain and cognitive reserve might play a role in moderating the link between type 2 diabetes and the rate of cognitive decline. So these are pathways, these are broad pathways that we explored, and still work needs to be done to see if there are mechanisms or pathology related to the mechanisms that can actually explain the associations we've seen between these pathways. These are my acknowledgements. Thank you.
Srikanth, that's fantastic. I mean, there's a symposium in that talk alone. We could just spend the day talking about those results. It's very interesting. I'm sure the D2 people in this room are very interested too. But remember, questions at the end.